All right, in chapter 26, we are going to cover the circulatory system, talking about the blood as well as the lymphatic system. All right, so some of the unicellular protists, they still need oxygen and nutrients, but they're obtained just by simple diffusion. So moving from a high to low concentration from their environment or to the environment. Then the cnidarians and the flatworms, their cells are either directly exposed to the external environment or to some body cavity that functions in digestion, and that's referred to as the gastrovascular cavity. So in vertebrates, the circulatory system has three main functions. Transporting, so that's providing nutrients and gas exchange of substance essential for cellular functions are taken there by the circulatory system. It's also very important, though, in regulation, so temperature regulation, particularly using the countercurrent heat exchange. So again, our blood flow, we get flushed, or we can, whenever we're hot, that's the blood going to the skin to release heat, but not so much whenever we're cold, when we're keeping that heat close to the internal parts of our body. And the cardiovascular also is involved in protection against injury, but mainly that lymphatic system we'll talk more about from foreign invaders or toxins that can be introduced. So this countercurrent, again, heat exchange that we see here. So again, that the warm blood versus the cold kind of blood is going to keep that core body temperature about the same. So those arteries and veins, so the arteries are kept in the center and the veins are kept on the outer side, and that's going to keep the core of that organism at a relatively normal normal temperature. So we look at what this actually looks like. So the circulatory system has three main parts. It has the heart, which of course is the muscular pump that pushes the blood through the body. The blood vessels themselves are the tubes that carry the blood, and then the blood is the fluid that moves through those vessels. So that blood movement is directional. So it moves in a cycle from the heart through a system of vessels back to the heart. So as blood leaves the heart, it moves in arteries, so arteries or away. And then those larger arteries pass into smaller ones called arterioles, and then to the smallest part, or the smallest vesicle, which are called capillaries, and that's then going to connect the arteries to the veins. So those small veins are called venules, and the capillaries are what's going to be in between. So they're where we're going to convert from the artery. So we go from the heart to the bigger vessels called arteries, then to smaller vessels called arterioles, and then into the smallest, which are the capillaries. And then that's where we're going to switch into the veins, the smaller tubes called venules, then into larger tubes called veins, and then back to the heart. And of course, the color change as the blood moves away from the heart, it is oxygenated. As it goes back toward the heart, it is deoxygenated. So these capillary beds, again, the smallest part, this is where they're very narrow, and the reason for that is to give a lot of cross-sectional area that goes from one blood vessel type to the other. So these capillary beds can be opened or closed based on the type of tissue. And they also have these precapillary sphincters that can contract or relax depending on what blood flow is needed as we move in there. But at the capillaries, this is where that gas exchange and exchange of metabolites is mainly going to happen, because those arteries or even the arterioles, the walls are so thick it's hard for anything to move out. So the capillaries is where we get the exchange. And again, just kind of showing this capillary action here. So we have the precapillary sphincter, in this case is closed, so it's preventing anything from moving into the capillaries for exchange. So if there's already enough or there's nothing to pick up, we close those off. or it can be open if they need to get oxygen or need to dump off carbon dioxide. So a little bit more about the arteries and arterioles. Again, the artery is just kind of a simple pipe. Needs to be able to expand to control against the pressure of the heart during contraction. So they have both elastic and smooth muscles, so they can change their diameter, the elasticity, but also that contraction of the smooth muscle. Then the arterioles are a little bit different, mainly because they're smaller in diameter, and they're going to respond more to the nervous and hormonal stimulation, more so than from the pressure that we see with the arteries. So they constrict or expand the blood flow based on stimulation from the nervous system or the endocrine. Okay, so again, just kind of looking at the layers here, the blood vessel. So the innermost part, we have that endothelium, so those little squamous cells that are nice and flat. They're simple, just a single layer. And then the elastic layer outside of that allows the diameter to change. Then the smooth muscle that provides that contraction when needed, and then ultimately surrounded by connective tissue for support. All right, so then at those capillaries, again, this is where that oxygen and food molecules are transferred, and then the cells will pick up waste in carbon dioxide. So they drop off one thing, pick up another. They are very narrow with very thin walls to allow that exchange to occur. So almost all the cells are no more than about 100 micrometers from a capillary. So they're very close to those capillaries to allow them to be supplied with oxygen and nutrients and dump off their carbon dioxide and waste. So because of that, the blood pressure is actually pretty low in the capillaries compared to the arteries. So our capillary structure, again, pretty simple. We just have endothelial cells here where we have that gas exchange. So those squamous cells that allow for exchange of nutrients or gases. Right, then our veins, these are returning the blood to the heart. So these are actually, of the veins are thinner because the blood pressure is decreasing. So less blood pressure as we move through. So they're a little bit thinner, and they are unidirectional valves that prevent the flow from going backwards. So since there's not a lot of pressure, we don't want it to like slide back. So those valves prevent it from going backwards. So kind of looking at their structure here, again, that endothelium that we have here, so those squamous cells, then the elastic layer on the outside, smooth muscle around that, and then the connective tissue around that. So very similar to what we had seen with the arteries. All right, so then as the blood is flowing through the veins, so again, our veins are here, the skeletal muscles are going to help contract. So as they contract, again, that valve is going to close, and that prevents the blood from flowing in the opposite direction. So the contraction of the muscles as well as the closing of this valve keeps everything moving in one direction. Right, now another part of the circulatory system besides the blood is the lymphatic. So this is going to be recovering some lost fluid. It's also, though, involved in our immune system. So the vascular tissue is actually pretty leaky. So from that capillary exchange, not only are gases and nutrients moving, but also water is moving through there. So we lose about 4 liters of fluid every day. So in order to kind of collect and recycle that fluid, the second kind of circulatory system is the lymphatic system, and it's going to help recycle some of those fluids. So then the fluid inside of that, instead of being blood, is called the lymph, and ultimately it just re-enters the bloodstream through the veins that are there in the neck, and then it again supplies that fluid that was lost back to the circulatory system in the blood. All right, you can see the lymphatic pretty much mirrors and parallels our circulatory system. So where we would see arteries and veins, we also have the lymphatic system because, again, it's harvesting and collecting those. Of course, we have these lymph nodes in lymphatic organs like the thymus and the spleen, as well as the vessels. And this is also why typically whenever if a tumor is being removed, they'll take a couple lymph nodes from the area to check those, because again, since the lymph is kind of harvesting some of those lost fluids, if that tumor has metastasized and started to spread, oftentimes that's the first place that those cancer cells would move to, or the lymph nodes closest to the tumor. So by checking those lymph nodes, it gives a good indication of whether or not that cancer has metastasized. So again, just kind of showing this reclaiming of the fluid. So again, we have the arterioles here coming into our capillary where we're getting exchange. And then we also have osmosis here, the movement of water. So again, we're having exchange here in the capillary part of our venule that's picking that up. But then kind of in this in-between, we have this lymphatic capillary that is also going to be taking up the extrastitial fluid, or the other fluid that didn't get reabsorbed in the capillary. So some of the functions here, again, 3 main functions, again, returning proteins to the circulatory system. So if it remains there in the tissue, though, that's what causes swelling or edema. So that's why if you have like a broken bone or a bruise or a sprained ankle or something like that, it's again because proteins have kind of gotten out of the blood whenever those blood vessels rupture, and those proteins induce the swelling because that's what the lymphatic system is meant to do. It also helps transport fats that are absorbed from the intestines, and it's where a lot of our immune cells are, so the body defense. So again, those lymph nodes there in those lymph vessels, so especially there at the spleen, this is where the bacteria and the dead blood cells actually get destroyed. And also the thymus is part of that produces the white blood cells that are needed for the immune system. All right, we also have blood plasma to talk about. So it's kind of a complex solution. Here it has a few different things in it. It has metabolites and waste that are there. It contains salts and ions like sodium and chloride or bicarbonate to help with keeping pH normal. And then a lot of different proteins that are in there. That's going to help keep the water in the plasma. So it creates, so we talked about osmosis and, you know, like hypo and hypertonic, keeping proteins there keeps the water kind of stays with other solutes like other proteins. So it helps keep the water inside. There are a lot of different types of blood cells. We won't be covering them all. Again, if you go on to an anatomy and physiology or a micro course, you'll be learning more about them. But for here, just kind of getting an appreciation. So 3 main cell types. We have erythrocytes or red blood cells. That determines what's called the hematocrit, which is how many red blood cells there are for each like liter or volume of blood. So it's normally about 45%. So about half of our blood is made up of red blood cells. And then we also have leukocytes or white blood cells that help with our immune system, and then platelets, which are cell fragments. So the erythrocytes, again, red blood cells, pretty common. We think about it kind of a flattened donut here that's indented. It has a bunch of hemoglobin that's packed in there, and its main job is to carry oxygen. It does not have a nucleus. Now, it's still considered a eukaryotic cell, but those cells have been denucleated, meaning the nucleus has come out, and they do not live very long, so they are regenerated very quickly, and they get synthesized in the bone marrow. Now, the leukocytes and platelets, they don't have the hemoglobin. That's why they don't have that red shape. So essentially they're colorless. Again, lots of different types of leukocytes like macrophages and basophils and all these types of cells that have different functions to help with the immune system. Again, their main purpose though is defending against those invading microorganisms or foreign substances. And then those platelets or cell fragments, They're just pinched off from larger cells in the bone marrow. They're called megakaryocytes, and they're mainly involved in clotting. Right now, it's kind of switching into the fish circulation. So the chordates are thought to be an ancestral organism to the vertebrates. They have kind of a simple tubular heart. And then, so again, of course, moving from the gills, they required a more efficient pumping for a true chamber pumped heart. So the fish heart essentially is just a tube with 4 chambers that are arrayed one after another. So it has the SV, or the sinus venosus, the atrium, are kind of collecting chambers, and then the ventricle and the CA, or the conus arteriosus, are pumping chambers. And then again, those are then reduced in the higher vertebrates. So those chambers contract to allow rhythmic sequences or movement of the blood through the heart. So then it gets pumped to the body, and it's fully oxygenated because it goes to the gills first, but it also has much less pressure than what a vertebrate heart would. So again, just kind of looking, it's the same basic idea, but again, we have these 4 units that are just in sequence to each other instead of like our 4-chambered heart. They just move from one to another in a continuous kind of line, but still carrying blood away and then coming back through those veins. Right now, if we take a little trip off the lungs, we'll come back to them in the respiratory, but they definitely tie into the circulatory. So as the lungs came along, this caused a major change in the pattern of circulation. So after the blood is pumped to the heart, then it goes to the lungs, but it doesn't go directly to the tissue. It actually comes back to the heart. So unlike what was seen in the fish, where it just kind of was continuous, it actually leaves the heart, goes to the lungs to get oxygen, and then comes back before it goes to the rest. So we have kind of 2 different circulations: the pulmonary circulation, which is the connection between the heart and the lungs, and then the systemic circulation is between the heart and the rest of the body. Now, amphibians and reptiles, the amphibian heart has features to prevent the mixing of deoxygenated blood from the body with the oxygenated blood from the lungs. So in the atrium, it's divided by a septum that separates the blood. So it keeps the oxygenated away from the deoxygenated. So then there's just one single ventricle, but again, little mixing occurs because some species of amphibians have these folds in the ventricles that direct the flow of blood to the atrium. And then that conus arteriosus is actually branched instead of like what we had seen exactly with the fish. So with the amphibians that are in a water supplement, with oxygenation of their blood by obtaining additional oxygen across the skin, and this is called cutaneous, or like skin respiration. They actually breathe through their skin. And then the reptilian heart has additional specialization. They have a partial septum in the ventricle, and then that conus arteriosus is actually become incorporated into the large arteries that are leaving the heart. And then if we get into the mammals and the birds, with their circulations, they have a 4-chambered heart with 2 complete pumping circuits. This increases the efficiency of the double circulation in the mammals and the birds that may have been important for that endothermy. So they have a more efficient circulation that's necessary for their higher metabolic rate that's required. So then their heart, the oxygen-rich blood returns to the lungs through the pulmonary veins, goes to the left atrium of the heart, then it flows passively through the mitral valve into the left ventricle. Then the thick-walled left ventricle contracts, sends that oxygenated blood through the large artery to the aorta and then out to the body. And then that backflow is prevented by the aortic semilunar valve. So that's what we have in the mammalian heart. That's what our blood flow looks like. So then the blood travels through the blood and returns to the heart via the vena cava, the largest of the veins. That's going to flow into the right atrium. Then it flows through the tricuspid valve to the right ventricle. Then the right ventricle contracts, pushes the blood into the through the pulmonary valve into the pulmonary arteries that then go to the lungs. So if we look here at the heart and just that circulation, so it's important to understand the blood flow. So as deoxygenated blood comes from the lower or the upper parts of the body, we come into that vena cava, it comes right into the right atria. And then it moves through the tricuspid valve into the right ventricle. And then as it moves through there, it's going to flow into the pulmonary artery. And then it branches, of course, to the right into the left lungs. Once it gets oxygenated, it's going to come back through those pulmonary veins. So this is the exception. The pulmonary veins, typically veins are not oxygenated, but the pulmonary veins are. They come into the left atria of the heart. They're going to go through the bicuspid valve and into the left ventricle, and then they're going to flow into the aorta to go to the rest of the body. So again, most veins are deoxygenated, most arteries are oxygenated, but the pulmonary artery is not oxygenated and the pulmonary veins are. So they're opposites. All right, so then just looking again within the mammals and the birds, again, that circulation from the heart out the aorta through the different arterioles to the capillary bed. So you can see various capillary beds shown here where that gas exchange happens. Then that vena cava brings everything back to the heart to go to the lungs to become oxygenated. So now looking a little bit at the heartbeat that we get. So of course that lub-dub sound that we hear is the closing. So the lub that we hear first is the closing of that bicuspid and the tricuspid valves at the start of the ventricular contraction. And then the dub is the closing of the pulmonary and the aortic valves at the end of the contraction. And then a heart murmur can sometimes occur that's due to some turbulence that's created by the valves not closing completely. So it creates more of a, instead of just a lub-dub, it's more of a turbulent kind of vibration sound. All right, then our blood pressure is one of the things to examine. So the diastolic pressure that we get is the low pressure. This is whenever the atria are filling. So as blood is coming into the heart, as they are filling, that's the diastolic pressure. And then the systolic pressure is the higher pressure, so the higher number on our blood pressure. And that's as it occurs as the ventricles are contracting to push the blood out of the heart. So measuring of that, so of course using the stethoscope, so that blood pressure gauge is applied to increase the pressure, and then that first sound, that first pulse sound that occurs is the systolic pressure, and then once they're no longer able to hear the pulse, the kind of the beating of the heart whenever it stops, that's the diastolic pressure. So in between those, that's whenever you can actually hear the kind of pumping or the beating of the heart. So whenever it starts, that's the systolic. When it ends, that's the diastolic. So then we also have a couple nodes here to talk about. So the contraction consists of carefully orchestrated series of contractions that occur. So first the atria contracts, then the ventricles. So as it's kind of changed over time, what we get is kind of this pacemaker effect that occurs in the sinus venosa. And this is where that heartbeat actually originates. So this chamber is very reduced or very small in amphibians and reptiles. In mammals and birds, though, it exists only is the tissues in the wall of the right atria. It's called the SA node or the sinoatrial node. So it's the pacemaker of the heart that determines the rhythm of the heart's beating. And then the AV or the atrioventricular node, this is the contraction of the atria is initiated by the SA. So again, the membranes depolarize. We'll come back more to this when we talk about the nervous system. But this electrical signal passes to all the heart muscles in the atria. So then that wave of depolarization doesn't immediately spread, though, to the ventricles, because first it has to go through the cardiac muscle. And that muscle there is called the atrioventricular node or the AV node. And so this delay is about 0.1 seconds until the atria is actually finished contracting. So that electrical activity of the heart, so the ventricles finally contract after the signal passes through that AV node. So again, it starts in the SA and then to the AV into the atrioventricular bundle, and that's called the bundle of His. And then these bundle branches divide into the Purkinje fibers, which initiate almost simultaneous contraction of the right and left ventricles. And then, so that electrical activity can actually be measured. That's what you see on an EC or an EKG, which is that electrocardiogram. And it looks a lot like this. So with this ECG, so we have these different waves, again, typically about a second between this contraction that occurs here. So the P wave that we get here that's indicated down here. So again, this is as the SA node. We come in, we get filling of the atria here on both sides. So our atria are filling. That's the P wave. And then the QRS wave, which is this kind of blip down and then this peak up. So first we have this bundle of His here that is going to occur, and then our Purkinje fibers, that's going to allow that contraction that then allows everything to leave, or that contraction then of the atria to force the blood out of the heart.